So I'd like to get started. I'm Dr. Bert Eisenberg. I'm the Cancer Center Director here at Hoag, and it's my pleasure to kick off this program. At Hoag, we have a fairly robust molecular precision medicine program centered mostly here in the Cancer Center. And part of that program involves education. So this is the first in a series of seminars into precision medicine, and I want to remind everybody that this is Part A. Part B will be on Thursday, February 7th, in this room starting at 6 o'clock. I think you'll find that they're complementary, but not redundant. So I'm going to start by having our Director of Precision Medicine here in the Cancer Center, Dr. Mike Tumor, come up and provide the introduction.
Thank you. The first part of what I wanted to do is to just talk about, when we talk about precision medicine, what we're talking about and a little bit of the rationale. And of course, this is a new area in general, but I think we're all faced with the direct-to-consumer kits now, and so obviously patients are going to be asking you about it and talking to you about it, and the like. So, yeah. Again, as Bert said, this is really a cancer center initiative, and so a lot of the program will feature cancer, and we are doing better. I remember Richard Nixon, and it was 1968, declaring war on cancer. So 50 years ago, where are we? Well, we are making progress. This was just very recently published in Cancer this month, where it says that if you extrapolate predicted cancers versus the actual cancers, that that we're really are doing better. And that's multifactorial. Some of it's early detection, prevention, better interventions and treatments as well. So it's multifactorial, but I guess that's the good news, is we are making progress. We've got a lot of ways to go, and precision medicine will hopefully help us make more progress. What we mean by precision medicine is the application of all the technology of genomic sequencing learned by doing the human genome sequencing and all the analytic tools that were developed for that, but applying it to the care of patients, individual patients. And by that means we can identify and mitigate the risk of diseases, including cancer. Example would be identifying the BRCA gene mutation that Angelina Jolie made famous, but to identify patients at increased risk for breast, ovarian, pancreas, and prostate cancer, for example. To aid in the diagnosis and treatment of diseases, and also using genomics to select, in some cases, the most beneficial medicine or the safest medication, or one that has might be a safer or more accurately dosed medications. So, of course, our former president said, when he talked about the initiation of a program devoted to precision medicine, or that Joe Biden was spearheading, so what's the promise of precision medicine? Delivering the right treatment at the right time to every patient, every time to the right person, and for a small growing number of patients. The future is already here. So we are doing that, and we'll highlight some of the areas where we are. So one of the drivers, or one of the major drivers of precision medicine, is just this graph that says the cost of sequencing is going down. So, if it would cost, $1 million to sequence the human genome, it just wouldn't be practical. Plus, if it, took 10 years and $3 billion, like the first human genome, it wouldn't be practical for patients. But we're now talking about genomes under $1,000, and I was reading an article about, we're all using Illumina now as the second generation, reading about third generation sequencing, and talking about getting down to $100 genomes. So the sequencing costs are coming down, and also the speed to do it is coming down. So when you have a patient with cancer, you can't wait for a year or 2 years to get a sequence back. It's got to be done quick enough that we can act on the findings. So the other things that when you talk about genomic tests, payers have to pay for it. And the things they look at in particular criteria, but also just we as physicians need to know that tests have analytic validity. In other words, does the test predict either the presence or the absence of a particular genomic variant? How well does it do that? If there is a mutation in a gene, do you accurately find that? If you say it's not there, is that accurate? So the test has to be an accurate test or have what we could say analytic validity. Clinical validity, how well does the tested for genetic variant related to the presence or absence of risk of a disease? So in other words, if you have a mutation or an abnormal gene, how well does that predict the disease that it's associated with? Or, what way is penetrance, but is it clinically valid? And then lastly, clinical utility. And that's where we're still working in a lot of areas in precision medicine for the area of clinical utility. In other words, does this help the healthcare provider, the physician, better diagnose, treat, manage, or prevent a disease? So, if it doesn't help you do that, then, why do it? So again, insurance companies want to say that they're not going to pay for it unless it has clinical utility. And this is the area where we're still working to get that information. Now, another way of putting this is I got my MBA some years ago, and this was a graph that we learned about when you're looking at market penetrance, even an iPhone or something like that, that you have to jump this chasm. Some people will be early adopters and innovators. They want to try something because it's shiny and new, and I like shiny and new things. But before you get widespread adoption into the marketplace, you have to cross this gap or chasm right here. And that is where you get that is when people understand how it helps them in their day-to-day activities. Whether it's the iPhone, it's great because I can make phone calls, I can listen to music, take pictures, whatever, so I want this thing. But with tests, it's the same. Does it help me as a physician take better care of my patients? And when I can see that happening, then I'm going to use the test. So part of what we're doing here is to improve the understanding of clinical utility is we need to educate ourselves. None of us were involved in sequencing in medical school, except maybe Dr. Braxton and some of the younger faculty here. But the rest, when I went to medical school, none of this was being done. And so we have to, just like every other field of medicine advancing rapidly, we have to stay up on that. We have to better integrate data so that we get the outcomes linked to the genomic test and the treatments based on the genomic tests. And then we are using Synergy, and we'll show one slide a little bit how the new genomic technologies is driven by the fact that we have also targeted drugs that are able to address these genomic variants so that if we identify something, we have a drug then that goes with it that can provide benefit to the patient. So all that utility will then help drive the adoption of genomic testing. There's lots of uses, and this is application of different information we get. We have normal and germline. So we have germline, which is inherited mutations you could pass on to your kids. And this is the BRCA and all that. But there's increasing understanding of various genes that can confer increased risk of cancer. In some cases, like the red oncogene, it might be 100% chance of getting medullary thyroid cancer. And other times we have low penetrance, so you may have a mutation, but it only slightly increases your risk of tumor. And how we deal with that may be different because it may be surveillance versus prophylactically taking out somebody's thyroid. So the consequences of that surgery also make a difference. And then we have pharmacogenetics that affect genes that we have may affect how we metabolize drugs. So we can use that information. If you have a deficiency in DPD, for example, you can be at increased risk for very toxic effects of drugs like 5-fluorouracil. So, but then we also have genes that are mutated only in tumors. They're in the diseased tissue. So in a cancer, you can have acquired mutations in that cancer. And that can be used to confer prognosis. So we can predict that it's a bad-acting tumor because it has a certain set of genomic variants. Or we can use it to assess which drugs might be more effective for that patient. And in some cases, certain hematologic malignancies, or lung cancer things, it's really become standard of care to do that to guide therapy. So the benefits of genomic analysis in cancer is to secure a diagnosis in some cases. The presence of a genomic variant will help pathologists or make a diagnosis. We can predict prognosis, I said, predict drugs that would be indicated. If you have an ALK rearranged lung cancer, crizotinib or other ALK inhibitors or first-line treatments, in some cases better than standard chemotherapy in terms of outcomes with less toxicity. Vemurafenib and BRAF mutated melanoma, but you can repurpose these drugs to, for example, rare cancers where you can't do a study of 300 patients getting treatment A and another 300B. We repurpose for the same mutation in a rare cancer, and you have a drug that was developed for a common cancer, that mutation, you could repurpose that drug. And for example, an Erdheim-Chester disease where, rarely rare tumor, vemurafenib, the same drug, the melanoma drug, was approved for that based on, I think it was 17 patients, and having a fairly remarkable response. So you have the ability. And then other times you can predict drugs that you don't want to give. EGFR-directed therapy in KRAS-mutated colon cancer might be an example. So as costs continue to decrease, in contrast to other rising costs of treatment, so sequencing becomes more and more tractable. And then again, meta-analysis overall show improved survival with decreasing costs when we apply genomics to treatment. And that's an area of continued need for research and outcome data. But we've had transformative advances in oncology already due to genomics. We talk about one of the really early ones is imatinib in c-KIT mutant gastrointestinal stroma tumors, or CML, with the BCR-ABL fusion. Mentioned crizotinib, but in other lung cancers, EGFR, ROS1 inhibitors in lung cancer, BRAF and MEK inhibitors in melanoma, HER2 new directed therapy in breast cancer, sonic hedgehog pathway inhibitors in metastatic basal cell cancers, remarkable advance. Other conjugate, antibody drug conjugates in Hodgkin's and anaplastic large cell lymphoma. As I said, we have RET inhibitors in medullary thyroid cancer and other RET rearranged tumors. So there's been a lot of advances, and they continue to come. The pace of, until the government shutdown, the pace of drug approvals have been increasing over the last few years. But let's just take lung cancer, where it's probably the prototype, if you will, of having genomic testing as far as upfront testing in metastatic lung cancer. It's now the standard of care. But you have this array of different gene mutations, rearrangements, amplifications, and different aberrations that in this panel of genes, now you may see the prevalence for each one is low, 15%, 3 to 5%, 3%, 1%, but if you have one of these and you're the patient, you have derived remarkable benefit from the targeted drug. So in aggregate, you're getting up a fairly large number of large percentage that you may find these. And now some of these will correlate with other phenotypes, non-smoking Asian women, for example, things like that, having characteristic gene mutations. But how are we doing overall? And this is where the clinical utility that I mentioned before, still the outcome data is needed, but we have some. There's a phase 2 study that was published a couple of years ago in Journal of Clinical Oncology. This was a meta-analysis of 570 phase 2 trials, single agents, and over 30,000 patients. And what they looked at was patients that got directed to drug therapy based on gene mutations or their tumor profiling versus sort of the traditional non-personalized medicine approach. And when you had what we call the personalized or precision medicine approach, and that was used for the patients that had a higher overall response rate, 31 versus 0.5%, better prolonged median progression-free survival of 6 months versus 3 months, and overall survival of over 13 months versus 9 months, all highly significant. And the non-personalized targeted arms had poorer outcomes than either personalized targeted therapy or treatment with cytotoxic agents. So overall, personalized medicine arms using a genomic marker had higher response rates, better median progression-free survival and overall survival compared to personalized medicines, either using protein biomarkers, and it was also associated with less treatment-related deaths. So in general, the targeted therapies are overall more effective, but also less toxic. But this is the other area that we've had is the immuno-oncology, which has been an incredible advance the last 2, 3, 4 years of in treatment of a lot of increasing array of malignancies. And so the genomic predictors can also help predict response to these immunologic agents, expression of PD-L1, what we call mutational burden. If a tumor has a lot of mutations, doesn't necessarily matter what they are, but just a high burden of a lot of different mutations, this will suggest that you are more likely to get a response to the immuno-oncology agents. And also when you have defective DNA repair mechanisms that we can determine by sequencing, that you'd be more responsive to immunologic agents. There are new targets and novel targets coming up, and new testing is going to be on the, so far we've talked about everything doing DNA. Well, RNA is the next thing coming up, and we were just talking even yesterday with a vendor talking about transcriptome or RNA analysis coming out as part of the panels, and so there's increasing interest in that, and that may predict the expression of neoantigens, which can be used to help select immuno-oncology-based agents. One of the areas that's emerging is combining chemotherapy or traditional cytotoxic agents or targeted agents with the immunologic agents for increased efficacy. And down the road, which is still pretty much, I'll just say, it's not, maybe a little more than a pipe dream, but it's kind of around the corner, is precision medicine or individualized based vaccines and CAR T therapy, which is sort of directed, engineered white blood cells, for want of a better word, of trying to use the immune system and program the immune system to be able to target tumors. So who should get tested right now? I mean, there's still a little bit of debate, and obviously not everybody agrees, but there was a think tank called the Center for Medical Technology Policy that said basically everybody with stage 4 lung cancer, newly diagnosed carcinomas of unknown primary, stage 4 rare or uncommon tumors where there is no good standard therapy or the clinical guidelines or pathways that are relevant or helpful. A newly diagnosed stage 4 cancers where median overall survival is poor. That may be a little bit more controversial, but especially when they haven't responded to standard therapies. Stage 4 patients or patients with stage 4 tumors have good performance status, but they've exhausted guideline-driven therapy to find out what's the next best option. And hematologic options with limited treatment options and defined clinical care guidelines. So I mentioned a little bit also pharmacogenetics. I'm just talk about that. There's increasing understanding of the effect of individual genetics on our ability to metabolize drugs and how effective drugs may be. A group called the PharmGKB, which is an NIH-funded resource, collects this data on patients and drug response. And they've identified information linking 650 drugs, 135 pathways, have issued 101 dosing guidelines based on drugs, and incorporation into drug labels information about genetics. A very well-known one is and this is directly from the FDA label for warfarin, 2 genes which affect the ability to metabolize warfarin. And if you have the variants, so VCORC1 is the one gene, and of course the genetic code is G's, C's, T's, and A's. So if you have this particular variant, and then look at this other gene, CYP2C9, and if you have the allele type 1, 2, or 3, and the mix, so then they issue recommended starting doses. But if you have, for example, 3 and AA, you're very, very sensitive to warfarin, so you would start on a much lower dose. And this has been shown to decrease the bleeding complications and increase the safety of the drug. We also know that the hereditary or acquired deficiencies in protein S or cofactor protein S have been associated with increased complications like tissue necrosis. But we also know that we talked about this CYP2C9 is involved in drug metabolism of a lot of different drugs, And so if you have inhibitors, or if you're on drugs that are inhibitors, then you can increase the effect of warfarin. And if you're on inducers of that same gene, you can decrease the effect of warfarin. So it's important to look at drug interactions based on how these affect. And here's a partial list, if you will, of drugs that it would affect. It's part of the cytochrome P450, but if you look at all these different drugs, capecitabine, amiodarone, different medications, meconazole, that would affect inhibitors and inducers, rifampin and phenobarbital and others. So important to know that around the genetics, and it can affect drug safety. So just in the last couple closing remarks, we have a team here, and this is just some you'll hear from much of us today. Our genetic counselors are in the back here, and Genio is the head of that group will be speaking with us, but they'll introduce others as well, and they're all representing our team, and there's a brochure here with that picture. So what do we offer? We have molecular tumor boards. Second Thursday of every month at 7:30 in this room. So everybody's welcome to come to that. We discuss patient cases and the genetics related to it. And we also support the specialty tumor boards when molecular profiling is done. We review all the molecular profiling of cancer samples that are done here, and I help working with the team of genetic counselors and all that. We do look at individual reports at the request of physicians or oncologists in particular to help interpret and provide additional guidance and information based on the sequencing reports to identify other treatment options or clinical trial options, educational outreaches we're doing now, support research initiatives, and also some of the foundation is represented here also is in the fundraising efforts. Tumor boards, as I mentioned, we have the variant scientists, who you'll be hearing from in just a little bit, is on our team, and we present at the conferences. We look at the case history, all the imaging, pathology, and the molecular testing, and then highlight particular journal publications and drug approvals that might be relevant to that particular patient. So far, we've done 291 tests over the past 6 months, working with Caris is our primary sequencing vendor, but of course we look at other vendors as well, such as Foundation. Presented so far 23 cases at our initial tumor boards, seen additional 94 consults, made additional treatment suggestions based on sequencing on 93, and in some cases we'll look at the gene mutations and say, well, this could be a germline mutation and recommend referral for genetic testing and genetic counseling. So we've done that out of 291, 36 patients. So what do you need to know, and this is what we're going to get out of this course today, a little bit about the basics of genetics and genomics, sequencing technology, which we'll cover more in the next session in 3 weeks' time, understanding the various tests. They do have differences and they do have limitations, how variants are interpreted and characterized, how the genetic variants and risk confer risk to patients and their offspring, and how we match drugs to genomic variants. So again, we'll be talking about these various things in the next. My next session will be talking about some of these particular the test and sequencing technologies. So now, take a few minutes for questions. Anybody has questions? All right. So we'll move on to Sarat Darabi. She's a newer member of the team. I think just started a few months ago, and she is a PhD scientist that joined us from Ambry. Does germline genetic testing. She had her PhD in Clemson, so I was teasing her she should have worn her football jersey, I guess, but her PhD in healthcare genetics at Clemson University, and and we're very fortunate to have her. She does review with me all the the genomic tests of tumors that are done, and so very often we're here for that, but she's also our lead presenter for a lot of the molecular tumor boards. So, Sarat, thank you.
Hi, thank you for the introduction, Dr. Tumor, and thank you so much for showing up in the rain. I think the rain stopped, but appreciate you all coming. My part of presentation is, I get excited for, but is basics. It's just a refresher. We try to make sure we are ready for the next presenters. So we'll go over some terminology and some basics review, and then we move on with the next presenters. We'll define the basics of genetics and genomics, the structure and function of human genome. We'll talk about a little bit oncogene, tumor suppressor gene, and a little bit talk about epigenetics, not a lot this time, but we will cover that in another talks. This is human karyotype. I don't know how many of you did karyotyping. You know that it's not going to be this beautiful. This is the most beautiful one that you could pick. So the one on the left is a male. So the karyotype is a picture of all chromosomes of one individual. So on the left is a male. This is a Y chromosome, and this is a X, so XY. 46 chromosome, XY, this is how we name them. 46 is a total number, so there are 23 pairs, one from each parents. And an XY means that this is a male. And the one on the right is the female, has two X's and no Y. And they all sort out by their size or a different specific other concept that they divide them. But then you can see them in the size, and they can see abnormalities, and very hard to find those. And they are different banding techniques that they use to be able to actually visualize chromosomes under a microscope. Genetic information. So in general, DNA is like a twisted ladder. We call it double helix. One part of DNA called gene is a region that encodes for a functional product. That could be RNA, it could be protein. We have about 22,000 genes in the genome, and a lot of them are not known, their function. However, the genome is a complete set of the DNA, the entire genetic information called genome. And the chromosomes are composed of DNA and protein. They all located in the nucleus. We have, as I said, 23 pairs. They are packed in the nucleus. So this is a chromosome. Not all chromosomes look the same. This is just a typical figure for the presentation. There is a short arm, stands for P for petite, and there's a longer arm, and we call it Q. Does anybody know why we call it Q? Yeah, it just comes after P. So they call it Q. So what is DNA? DNA stands for Deoxyribonucleic Acid. It's a double-strand chain of nucleotide. So we have, as you can see here, it has three components. One is a sugar called deoxyribose, and when we go to RNA, we see that is a ribonucleic, so it has an oxygen here. So the reason they call it deoxy is because there's no oxygen here, there's carbon. We have, as you can see here, there's a phosphate group and a base. So the base, we have 4 bases for DNA, adenine, guanine, thymine, and cytosine, and we take the first letters and we call them AGCT, and we call them AGCT now on. There are some rules for base pairing, and these are important rules, and if they're not followed, then there is a mutation, there is a variation, and something might go wrong. As we talked about the ladder, the side of the ladder of the DNA is phosphate and a sugar, and they are held together by hydrogen bonds. And we have different units. We call it base pair, or kilobase, or megabase, depending on the size. Example here, this is 4 base pair. DNA sequence, what is DNA sequence? We talk about it a lot, this terminology. Order of nucleotide along one strand. Like all this in one strand, they call it DNA sequence. Example I have here, this is part of BRCA2 gene, just a little part, and it has all this ATCG sequence that we can see. So we talk about sequence, we talk about base pair, kilobase, megabase a lot throughout our presentations. Now to RNA. So we talked about it. The sugar is different here, and the base is different than DNA. So the base here for RNA is uracil, but everything else similar to DNA. I wanted to touch on telomere a little bit. I'm not going to go deeper on this. Dr. Dalili has a couple of slides on telomere coming up. But the tips of the chromosome called telomere. We hear about telomere in aging, in cancer, or other diseases. What is it for? Is to protect the end of the chromosomes. If the end of the chromosomes are not protected, they're fused together. So in order to be kept safe and not fused together, there are certain repeats. There are TTAGG repeats several times at the tips of the chromosome. And every cell cycle, when the cell divides, they get shortened to the point that there's no more repeats left. So that's the time that apoptosis or cell death happens. Central dogma of molecular biology is the flow of genetic information, which is when DNA makes more DNA, called replication, or when DNA makes RNA, called transcription, or when RNA makes protein, translated to protein. So these are some codes in RNA. The codes are called codons. There are 3 codons for each amino acid. However, some amino acids might have several codons. As you can see in this table, I don't want you guys to be able to read that, but this shows the different codons that are in this table. So they code for protein. So if these codons are mutated or changed, then it might code for another amino acid. So the whole protein might change to some other protein or non-functional protein. So gene structure. So this is just the example of a short gene. Exons are the boxes in blue that are code for sequence, and they're expressed as proteins, so they are coding sequences. Whereas this introns are intervening sequences that they're initially transcribed to RNA, but they're not going to be in a mature mRNA. Promoter region is a sequence of DNA that is responsible to say that the gene needs to turn on or off. There are some regulatory elements here and there. I didn't put them all here, but they are regulatory for gene expression. How the protein synthesis is done. This is just a little simplified figure again. This is our DNA. Through transcription, through a process, and transcription and through processing, this RNA intron sections get spliced out. We call it splicing. So in an mRNA, this is a mature mRNA that introns are spliced out. They're taken out. And then when it's translated, it makes protein. And this part, human genetic variation. So all genetic variation originates from a process called mutation. We have several different terminology here. You talk to me, I might have one terminology used, and then you might talk to some other people. We tend to confuse people with different terminology, but we try to follow up the consensus American College of Medical Genetics guidelines, but sometimes even with us, we're trying to be using the same terminology. We might not come with a good one. So here I just go by my version. So genetic variation, genetic mutation, genetic alterations, they're all the same. We have here, divide them in two. Either structural variants, which is big changes in DNA. That could be from missing the entire chromosome or addition of entire chromosome. That would be as big as that. Or could be like translocations, smaller insertion, deletion, or copy number variants. The other group are single nucleotide variants, single nucleotide polymorphism, point mutations. They're all the same. They're single nucleotide changes, could be missense, nonsense, silent, or frame shift. I have examples of those in the slides coming up. This is an example of numerical variance. I'm not going to go over the details because Dr. Dalili has the slides for that, but this is shows. So the bigger the mutation is, less viable they are. So these are just a example of viable numerical variants. Because if they're like chromosome 1 missing or extra chromosome 1, we don't see it because they might not be viable. Chromosomal mutation. So when we talk about deletion, duplication, inversion, these are just the figures, examples to show you what we mean. A deletion in a chromosome is one section of chromosome can be deleted. And it's important. Location, location, location is important. Where that mutation happens, where that deletion happens. Exactly the same with duplication. So one section is doubled or inverted, or insertion of one piece of some other chromosomes to one chromosome, or translocation. Each one of them are important. The location, the genes are involved in that region. This is a table from our Thompson and Thompson genetics book, our Bible. They have different type of mutations as single nucleotide polymorphism, this is the same as SNV, single nucleotide variants, as one base pair. Insertion deletions are limited to one base pair to 100. Copy numbers are 10 kilobase to 1 megabase, and inversions can be different. For the copy number variants in different literature, different textbooks, you might find different numbers. So that's one of the areas that people have different standards for copy number variants and calling a copy number. Example of mutations. So they are compared with a reference. What is a reference? They sequence a lot of healthy individuals, and they go by their sequence of their genome, and they compare everyone else with them. This is a reference. We're focusing on this T here, TA here. So when the substitution happened, the T is substituted with G, so G, of course, it binds with C. So this is called substitution. Deletion is when the T is deleted, or insertion with another G is inserted here. What happens to the protein depends. These are the example of single base mutations. Here we have the reference or original DNA and an mRNA and a protein. So here we can see this is this codon codes for arginine, but for the missense mutation is one nucleotide mutation. This arginine is now proline, is another one. So this is a missense. Or it could be nonsense, which the mutation leads to a stop, which means the protein should be made, but it's stopped, so it's shorter. That's what's called nonsense mutation. Or there are another mutation. If you remember, I mentioned that there are different codons, could be for one amino acid. That's one of the examples. Although there is a mutation, but it still encodes for arginine. This is called silence mutation or non-synonymous mutation. Or I'm sorry, synonymous mutation. Frame shift is another one that is important for all of us. So this is normal DNA again, and an mRNA, the code of 3. If a GC is added, so is not divided by 3, that means that the whole 3 code is changed. So instead of making serine as an amino acid here, is glutamine. And it changed the entire polypeptide to the end for that insertion. That's called frame shift. I'm not going to go over the details here, but this table just shows the number of mutations are seen in diseases. So as you can see, most of the mutations are missense mutations, whereas the less percentage are seen in the large deletions. Somatic versus germline. We talked about this all the time, cancer, genetic testing, germline testing. So what are the differences? The somatic mutations only happens in non-germline tissue, whereas germline, it presents in eggs and sperm, and it can be passed on to the next generation. However, somatic mutations are not passed on to next generation. Cancer is fundamentally a genetic disease. So it could be either through activated mutations or inactivated mutations. Examples: when oncogenes are activated, or for inactivating mutation if it's the tumor suppressor gene or DNA repair gene. And these are the examples. I have examples in the coming slides. This is a figure, different versions is an analogy of tumor suppressor and oncogene. So for oncogene is like the green light, or you push the accelerator, it just say go, and then to divide to the cell, this is a signal, we need to divide. But then tumor suppressor is a brake, say no, we should not move on with the cell cycle, we should not do the cell division. Example of proto-oncogenes. So proto-oncogenes can be converted to oncogenes with different mechanisms. I have 3 examples. Point mutations can cause an overactive protein. The amount of protein is the same, but the protein is hyperactive. Whereas when it's amplification, there are several copies of one gene. So we have several proteins, but they're not overactive, they're overexpressed, more protein made. Or it could be due to fusion, and that fusions could active and make the gene products that they're hyperactive. There's an example of gene fusion in melanoma. BRAF is fused with the cytoskeletal gene, VIM. Exon 9 on one gene is fused to another exon 9 on the other gene. It doesn't necessarily need to be same exon. This could be exon 10 or could be 5. Is very random, but in this case, 2 exon 9s are fused together. Here's the BRAF, and this is exon 9 located. Where again, location is important. Where this is a breakpoint here for BRAF is where kinase domain is. For VIM gene regulation of gene expression is where the exon 9 and before are for. So when this gene is fused, these genes are fused together, the expression part, regulation of expression part of the VIM gene is fused to kinase domain of BRAF, so make it hyperactive. BRAF is a kinase, and the fusions will activate the MAPK pathway, and for specific BRAF fusions, the selective inhibitors are not effective, so suggesting the MEK inhibitors would be beneficial for these patients. Another one is an example of tumor suppressor genes. P53 is an example. This is an example of inactivating. So what P53 does when a DNA damage happens, abnormal cell cycle. So what P53 does, arrests the cell cycle, stop the cell cycle till that DNA is repaired. And then when the DNA repaired, then the cell cycle can restart. But if the DNA is not repaired, then there's a signal for cell death or apoptosis. However, when the P53 is mutated, we see a lot of cancers with P53. When it's mutated, when there is a damage in the DNA repair, the cell cycle continues. So we have the cells that they can become cancerous. DNA repair pathways is another one that inactivating mutations can cause cancer. I'm not going to go over any of these now here, except for one. Dr. Braxton have more in-depth presentation on this. But I just wanted to point out homologous recombination for BRCA1 or 2. We talk about PARP inhibitors, and I just want to touch on the mechanism there. So this figure shows here is a DNA that has a single strand break. For the single strand break, usually the mechanisms are to come and then repair the break, and then DNA is repaired here. But when PARP inhibitors are involved, this repair mechanism will not be able to work. So what happens, this single strand will be double strand break. Now it's time for the repair mechanisms that have double-strand break repair, which is a homologous recombination, on patients that they are deficient on a homologous recombination, like the patient with BRCA1 or 2. So when it's double-strand break, they cannot repair this, and then it goes to cell death. So that's the theory about behind the PARP inhibitors for these patients. Epigenetics. As I said, I just touched on it for just a couple of slides, and is yes, is going to be a little complicated, but we will have epigenetics for later seminars. Different mechanism in epigenetics. What is epigenetics? Is the changes in a gene function or gene expression is not to DNA sequence change. So all the mutation and variation we talked about are sequence changes, but epigenetics is not sequence changes. It's a change in a gene expression. There are different mechanisms, but I will talk about DNA methylation that we all see in cancer. So methyl group is added to the cytosine. That's what's called methylation during cell differentiation. And what happens if the promoter, we talked about the promoter of the gene, but promoter is methylated, the gene is silenced. So the gene is not, the gene is silenced and is not expressed, whereas it's unmethylated, the gene is transcribed. So we see a silencing in tumor suppressor or DNA repair genes that it leads to cancer development and progression. One last slide. MGMT is an example as a DNA repair enzyme is affecting the efficacy of chemotherapy, but when methylated, it leads to epigenetic silencing, loss of protein expression, and then it leads to accumulation of the DNA damage that they're supposed to repair, and then increased sensitivity to temozolomide. For the next part, I will have introduction to clinical assessment of variants. I will show ACMG guidelines on variant interpretation and classification, and hopefully we will be able to see everyone in 3 weeks for part B.
Thank you, Sarat. I think we always learn every time you present. So next up on our program is Jeannie Homer. Jeannie is the lead of our genetics counselor group. We're very fortunate to have three really superb genetic counselors here in the Cancer Center. So Jeannie, I didn't know that. Went to college and studied Russian language, and then but she's later became a genetic counselor, and she got her master's degree from UC Irvine and became a board-certified genetic counselor, and has been here at the Hoag Cancer Center for the past 17 years. So she is going to overview our hereditary cancer program at Part A and go from there. So thank you, Jean.
Nice to see you all here. So this isn't working. Okay, here we are. So I've been asked to talk about who are genetic counselors and our hereditary cancer program here at Hoag, when it's a good idea to refer someone to see us in the hereditary cancer program. And then I'm going to go through a typical appointment from referral to the final report so you know what we're doing here. And then my favorite part is to give you an example of one of the wonderful families I've been able to work with here.
Can you do a little louder?
Louder? Is this better? Okay. So who are genetic counselors? We're kind of a misunderstood profession, but we're healthcare professionals with expertise in basic science, medical genetics, epidemiological principles, and counseling theory. And we've been involved in precision medicine since long before the name was invented. There are about 4,000 genetic counselors in the country, and we have different areas of specialization, including prenatal, pediatric, laboratory, cardiac, and cancer. And there are about 400 cancer counselors in the country, specializing just in cancer. And So the training, someone sitting at my table said, what do you do to become a genetic counselor? Well, there are very few training programs because obviously there are few genetic counselors, and I went to the UCI program. There were 4 positions accepted from 80 applicants, and I was lucky enough to be one of them. And then it's a master's program, 2 years master's program, and then we do our board certification, just like all you do. But our boards are the genetic boards, with the same boards that the medical geneticists do and the PhD geneticists, and then there's a genetic counseling board as well. And in my year, which was about 20 years ago, these boards were offered every 3 years, cost over $1,000, which was pretty expensive 20 years ago, and were offered in 3 places in the country, 1 on each coast and 1 in the middle. And so I studied for a year because I didn't want to not pass and have to wait 3 more years and cost all that money and take it again. So I was fortunate to pass the first time. And genetic counselors are also licensed in California, and then we maintain our certification and licensing through continuing education, which is really important in my field because things are changing daily and weekly. The genetic counseling field has a number of challenges, and I'll be mentioning a couple of them as we go. But the first one is really our name, counselors. People think, what's a counselor? Or they think of a different idea of counseling. So at a social engagement a few years ago, I met a physician, and I told him what I did, and he said, oh, you know, my sister was just found to have a BRCA1 gene mutation. Now, I don't need genetic counseling, but what would that mean for me? Can I have that too? And what would that mean if I had that? And how do I get tested for that? And what if I have that? What's going to happen to my health insurance? And what would this mean for my children? But I don't need genetic counseling. But that's exactly what genetic counseling is. That's what we do. When we meet with a patient or a family, we explain the genetics, we answer questions about genetics, we analyze the family tree, based on analysis of the family tree, we talk about, what is the likelihood that there's any kind of hereditary cancer going on in your family. We discuss legal and insurance implications, and I put a star there because I want to explain to everybody that there is a federal law called the Genetic Information Non-Discrimination Act, or GINA, that prohibits healthcare providers and employers from discriminating against someone based on their genetic information, which includes family history or genetic test results. There are some limitations to that, but we explain what those are. Genetic counselors then, if the person wants to do genetic testing, we figure out what's the best lab that fits with their insurance, that has the genes that they want to be tested for, so we arrange all of that. And when the test results come back, we go over those results with the patient. What does this mean for you? What does this mean for your family? And that's just a really, really, really important part because there's a lot of confusion and complication in genetic test results. And then one of the things we do in our program that's very time-consuming is to write this all out in a big summary report that goes to the patient and goes to any of the doctors that they want that to go to. So by being genetic counselors, I want to say we're not therapists. We're also not physicians in that although our license allows us to order genetic testing, we can't order MRIs or we can't do physical exams on patients. We're also involved in publications, presentations, and research. Now I just want to go over about our hereditary cancer program. It began in earnest in about 2002. And we were focused on hereditary breast and ovarian cancer associated with BRCA1 and BRCA2. And Dr. Dillman, who's in the audience here, was very much involved in setting up this program. And the purpose of this is to really, to do this precision medicine, to identify individuals so that their medical care, their health care can be guided by their genetic information. And so here's a picture of me counseling a fake person. This is a much younger version of me back in 2002. In 2002, that whole year, we saw 46 patients. In 2018, we saw 1,030 patients. In 2002, there were 17 physicians who referred to our program. In 2018, there were 225 physicians that referred to our program. And again, in 2002, it was all about hereditary breast and ovarian cancer, BRCA1 and 2. And nowadays, we see people for all different types of cancer, as well as other adult onset genetic disorders. And here's a picture of our team, and almost everybody in our team is here with our, number one cheerleader, Dr. Tumor, right in the middle there. When is it appropriate to refer someone to see us? Well, I think that anybody who is interested, concerned about their family history or concerned about what their cancer the implications for their family, should be able to come in for genetic counseling. A lot of people, where we look at their family and think, what the heck are they doing here? These are the most grateful people because they thought they were at super high risk, and they're so relieved not to be. Let me go back one second. And as I mentioned, we're going to talk about a typical appointment and later, and then an example family. But it surprises a lot of people to know that having some relatives with cancer is not the same thing as hereditary cancer. Out of all people diagnosed with cancer, this little blue wedge up here, about 10% is considered to be hereditary. And we have some clues to help us decide who's in that category. And one of them is young age of diagnosis. And when I say young age of diagnosis, I'm not referring to like children with leukemia. When I say this, I mean maybe being diagnosed 15, 20 years younger than an average person is for that type of cancer. So someone under 45 for breast cancer, someone 50 and under maybe for colon cancer. Also, a person diagnosed with cancer twice. And this would be someone diagnosed maybe with kidney cancer in both kidneys. It doesn't have to be at the same time. It can be years later. This is not someone like me with 4 basal cell carcinomas. That's from some other reason. And then another feature of hereditary cancer is a rare tumor or cancer. For example, paraganglioma, pheochromocytoma. Or sometimes it's a common cancer with a rare presentation. I saw a woman last week in her 50s. She had been diagnosed with 2 to 3 primary lung cancers, never a smoker. So that's kind of a common cancer, but rare in that person. Or a male with breast cancer is another example. The most obvious you would think of is multiple family members on the same side of the family in multiple generations with the same or related cancers. Now, related cancers are really just what the genes tell us. It's hard to know what's a related cancer if you're not an expert in this field. But Chelsea made up a little cheat sheet if anybody wants to take a picture of this. I mean, the common one that we know about is breast and ovarian cancer, but there's also colon and uterine cancer, and melanoma, and pancreatic cancer, and many more here. And just because cancer can occur in a family, it doesn't necessarily mean it's hereditary, so we have to rule out environmental lifestyle causes as well. And then one final hint about hereditary cancer is individuals of Eastern European or Ashkenazi Jewish ancestry have an increased hereditary risk for breast, ovarian, prostate, pancreatic, and colon cancer. So a typical appointment now. First of all, we see patients in both our Newport Beach and Irvine locations. People can be self-referred or can be referred by a physician, either through Epic or sending us a fax referral form, and then we'll take it from there. Insurance coverage is another thing that's misunderstood about genetic counseling. When I see people and I meet people during a social occasion, I'll talk about what I do, and they say, oh, that's never covered by insurance, is it? Well, genetic counseling is covered by all major PPOs and HMOs. It's not currently covered by Medicare, but my profession is working on that. And Hoag does offer cash pay discounts for patients who don't have insurance that covers Medicare. Medicare, sorry, that doesn't cover genetic counseling. Genetic testing, people think, oh, genetic testing is so expensive and it's not covered. And that used to be true, but it's covered by all major PPOs and HMOs when certain criteria are met. It's also covered by Medicare when criteria are met. And one of the laboratories that we use now covers genetic testing for all Medicare patients regardless of whether criteria are met, because they're trying to prove that Medicare criteria are too strict. So any Medicare patient now, we can test for 0. And for any other patient that wants genetic testing that may not be covered because they may not meet criteria, we can get them a test for $250 for up to 100 cancer genes. Before the appointment, we gather the family history either through paper and pencil, or more frequently now people just go to a link that we send them and they answer some questions about their families, and we end up with the family tree. So this is a family tree in genetics. Each row represents one generation. Circles are females, squares are males. The line through the symbol means the person is not living anymore. And I'm explaining this because in this talk and then next time, I'm going to go through a lot of family trees. And then something is inside the symbol if the person has a genetic condition. And for us, this is cancer that we're talking about. We also review medical records of anyone diagnosed with cancer. We want to know, they don't always remember exactly what type of ovarian cancer they had or when they had it, and so we review the medical records for each person before they come in. And we analyze their family tree for all known hereditary cancer syndromes or conditions. And for some of the conditions, we can do a computer analysis that will actually quantify what's the probability that this person has a gene mutation. We've already talked about the consultations. The consultations generally take around 45 minutes. But I had a couple patients earlier this week who had so many questions, and we met for well over an hour with them. And now I want to give you some information, just go over example family. And so this family, I call this the three sisters, and I saw them 15 years ago. And here's their picture. And just by the way, we really value privacy of our patients, but these patients signed waivers saying it's okay to talk about them. So these are the three sisters, and this is their mother. And their father's not in the picture because he has passed away. And that's why they came to see me, because he did pass away. And this guy, Phil, he was their father. This was a picture of the family taken in 1942 with all the brothers and sisters, and then the mother and the father. And then this is what the family looks like in their pedigree or their family tree. And here are the three sisters, Linda, Stephanie, and Rita. A couple of them have children. That was their mom you saw in the picture. And this was their dad. He had died at 69 of stomach cancer. And he had 4 sisters who also died of the same condition, and a brother who also died of stomach cancer, and the mother also. And one of the sisters got her stomach cancer at 25 years old and passed away. Everybody died within a year. 3 of them were in their 40s, and actually their dad, Phil, probably was really hoping, he hadn't thought maybe he was out of the woods, but he did get this stomach cancer at 69 and passed away. So of course, these 3 sisters are a little worried. We don't know what caused this. And there are several cousins, I think 17 in all. And Judy was one of their cousins, and she says, we don't know what this caused this. I don't know if I'm going to get it, so I'm just going to have my stomach removed surgically. And so I'm not going to get it. And that's what she did. But then what happened was the first person in the next generation got stomach cancer, David. And before he passed away, they had the sense to collect a blood sample and send it to British Columbia, where a group was working on trying to find the genetic cause of this type of stomach cancer. And after he passed away, they did find a mutation in a gene, And the cousins all got a letter explaining that. And these were the first 3 to act on that. And the gene is called CDH1, and it codes for a protein called E-cadherin. And so what we know is in their family, the people who had this condition had one of their chromosomes that had an abnormality or a mutation in CDH1 gene, and the other one had the normal version of the CDH1 gene. So this is their father's chromosome. We know that each of the daughters, the sisters, had a 50-50 chance to inherit one chromosome or the other one. And now we can have precision medicine because now we know what we're looking for and we can do a genetic test. So the implications of CDH1, again, it's not 100%. And when I saw this family back in 2004, there had been only 11 families reported in the literature with us. And based on those families, they knew that the females in those families had an 87% chance to get this stomach cancer. And 67% of the males got it. And the 10-year survival rate was 0. In this family, the 1-year survival rate was 0. And that women also have close to a 40% chance of developing lobular breast cancer. So in this family, the women didn't get the breast cancer, but remember, they died at 25 in their 40s, so they didn't have a chance to get the breast cancer. So I met with these sisters. 2 of them lived in Orange County, but the third one flew down from Seattle with her husband to go over all this, and we did genetic counseling and explained what the implications were and so forth. And then we collected a blood sample from them and we sent it off to Canada. And we waited about 5 weeks for the results. Nowadays we can get test results in much shorter time. But here were the test results. Linda carried the mutation. And she was kind of glad she was the one with the mutation because she didn't have any children she could pass it to. And Stephanie did not inherit the mutation. But Rita did inherit the mutation, which meant that her son and her daughter had a 50-50 chance that they were going to inherit it as well. So generally in cancer genetics, there are 3 options for people who inherit gene mutations. 1 is screening. 2 is sometimes there's medication that can be taken that can lower the likelihood of developing cancer, like tamoxifen with breast cancer. And then the third option is surgical. Now for this particular condition, it's called hereditary diffuse gastric cancer, screening doesn't work because the cancer is not a tumor. The cancer instead is something that spreads through the wall of the stomach, and so it's not seen as a tumor, and even if you do multiple biopsies, you can't always detect it. And there's no known drugs that you can take that can prevent this cancer, so surgery is the only option. And that's what these sisters opted for. And at that time, there is now, but at that time there was no surgeon at Hoag that did this surgery, prophylactic gastrectomy. So I found a surgeon in Seattle for Rita, and then for Linda, I found at Stanford a surgeon who specialized in this. So here's a picture of Linda before her surgery, eating a donut. And she sent me an email after the surgery and after Rita's surgery, and it says, I mean, they were going in for preventive gastrectomy, but she wrote the pathology for Rita found 2 small pockets of cancer in the walls of her stomach. My pathology indicated that the cancer had spread throughout the walls of my stomach but was fully contained. Dr. Norton removed more than 40 lymph nodes, which were all clean, so both of us are considered cured and are considered very lucky ladies. And here's a picture of Linda after her surgery. She looks very different. And she says, we're so lucky that we've had the opportunity to avoid this cancer. So what about the rest of the family? Judy, remember Judy had her stomach removed to prevent it? She didn't have the mutation. So not only do the people who are at risk benefit from knowing that to prevent cancer, people who aren't at risk don't have to do this stuff. ultimately, 11 of the family members had cancer, and this became news all over the country, all over the world. All these people had their stomachs taken out, and Hoag got a little mention in the news.
Now imagine this, a family of 11 cousins who all had their stomachs surgically removed, and it was done to save their lives. Paul Boyd has their story.
It's a joyous reunion for 11 distant cousins, some meeting for the first time. All share a common bond.
We don't have a stomach.
You see, all 11 cousins have had their stomachs surgically removed because all inherited a deadly mutated gene that predisposed them to stomach cancer.
It was like having a time bomb.
I was terrified.
Only 40 families in the world are known to have the mutated gene.
We got it.
Their cancerous gene first turned up 2 generations ago in their grandmother, Golda Bradfield. 7 of her 8 children inherited the deadly mutation.
My mom died at 49, stomach cancer. All my uncles, aunts died. I knew I was going to go.
And with that gene comes an almost certain death sentence. The disease usually strikes victims after they turn 50. And for women, the mortality rate is a stunning 83%. The shocking news set off a life-or-death chain reaction of phone calls and emails. The 11 cousins were spread out across the U.S. and all the way to South Africa.
It's brought us much closer.
Long time no see.
Incredibly, all 11 decided that having their stomach removed was the best chance for survival.
The choice was you either have it or you die.
All 11 successfully made it through the life-altering surgery.
We were all support systems for each other because it was a change, a big change in our life.
And there was one interesting change for the cousins. Many lost weight.
That's me.
Cousin Laura says she was once morbidly obese.
I've lost 125 pounds.
I lost 156 pounds.
So where does the food go? In place of the stomach, surgeons created a small sac using a piece of the intestine. Despite the surgery, the family remains upbeat and gathered at New York City's Tredici restaurant. They say they can eat anything on Tredici's menu, from steak and tuna to salads and vegetables. It's not what they eat, but how they eat.
When you don't have a stomach, you have to take very small bites and chew the food with each bite very well.
Without a stomach to soften food for digestion, they are supposed to chew their food 25 times, which Cousin Connie illustrates.
You spend forever chewing.
In the end, it was a celebration of life for 11 cousins, lucky to be alive.
Here's to us.
Oh, so another challenge for genetic counselors, and then just a couple slides left, is that, what we do is poorly understood. So I'm glad to have a chance to explain it. There's a lot of work ahead of time. There's a long time during the appointment, and then we have a lot of follow-up work. Here's kind of a big list showing all the things that we do. But what most people see is, whoops, that got in the wrong place. But it was a sample collection. They see blood draw. They think, that's easy. I can do that. So in Part B, we're going to give you more information about some of the wonderful families that I've worked with that have all the different syndromes that Dr. DeLilly will tell you about shortly. And then I think another interesting topic is, what about 23andMe and some of the other at-home DNA tests that we'll talk about.
It's great when we can prevent cancer. It's easier than curing it most of the time. So, our next speaker is Dr. David Braxton, who's pathology department. His specialty in pathology is GI, liver and pancreas diseases, but he also did a fellowship in molecular genetics. So he's worked with us on our high-risk protocols and genomic analysis, as well as some of the behind-the-scenes processes to get samples test out. Interesting thing I learned this week that was working with our vendor lab that we have a basically almost 98% success rate in sequencing tumors, which is terrific, and that speaks to the quality of the pathology that's done behind the scenes here. I mean, it's not easy to do that, but you can't send a good sample, you're not going to get good sequencing data. So kudos to our pathology department, but Dave and his colleagues have worked on that quite a bit. So he's going to talk to us a little bit about inherited pathology and molecular genetics.
Thank you for the lovely introduction. That's quite generous. So I have about 20 minutes to talk about the molecular pathology of cancer, and so that's quite a broad area to talk about. I really want to focus on DNA damage repair and what I consider to be fundamentals of targeted therapies. The goal of my lecture is to sort of hit the middle spot where the new genomics and their technologies of new genomics is overlapping with traditional molecular biology techniques and some clinical relevance. So hopefully I can hammer home some of these ideas here and make some points. The first thing to talk about is the hallmarks of cancer. A hallmark of cancer is anything that helps promote the growth in neoplasm and invasion, metastasis, all that. And genomic instability and mutation is one of the most important parts of that because dysregulation of the genome can promote any of the other hallmarks of cancer. So getting into DNA damage and the repair pathways, there are 4 major categories of DNA damage and 5 major DNA damage repair pathways. So any type of DNA damage can cause a base change. That's a single nucleotide variant. And so the first type of DNA damage to talk about is a single-stranded break where one of the phosphate backbones is broken in that DNA double helix. The base excision repair pathway is the pathway that repairs single-stranded breaks, and the PARP1 enzyme is active in that process. Double-stranded breaks is when both of those DNA backbones, those phosphate backbones, are broken. This is a very, very deleterious state for a cell to be in. It can cause a wide range of mutations, produce large insertion, deletion type mutations, copy number variants, and translocations. There are 2 types of repair pathways that can repair double-stranded breaks. In replicating cells, homologous recombination is active, and that's a very high-fidelity type of repair. Now, non-homologous end-joining is where non-replicative cells, that's basically the only option they have, and it's simply to take the ends of the DNA and stick them back together and try to fix it. That is not a very good way to fix DNA. It's particularly error-prone. Bulky adducts do form on the DNA bases here. Any chemical modification can change that base composition and form these bulky adducts and distort the helix. The nucleotide excision repair pathway is active in repairing those. And what I call replication errors, if you have an incorporation error during the replication of DNA for cell division, mismatch repair is the pathway that fixes those. And you can see C and T do not pair together here, and that's where you get mismatch repair. So the genomic consequences of failed DNA damage repair, I like to categorize these as really 2 major categories of genomic alterations. First is mutational. And a lot of the genomic studies that have come out recently have given us more criteria and sort of measurements so we can categorize them as mutator, hypermutator, and ultramutator types. Cancers that are deficient in mismatch repair are also known as MSI high cancers. Those are always a hypermutator type of cancer. Ultramutator type cancers happen when the DNA polymerase epsilon gene or POL-E is mutated, and those produce astounding rates of mutation in those cancers. The second general classification in genomic DNA damage is genomic instability. This is often associated with defects in P53 or P53 mutations, and this is categorized by genome doubling, where the whole genome doubles. You can get a lot of copy number variations, and you can get numerical and structural variants. So genomic instability produces large-scale abnormalities. And exemplifying this is the BRCA-mutated breast and ovarian cancers. They don't necessarily have high rates of mutations, but they have a lot of genomic instability going on. What happens when you have hypermutators or ultra-hypermutators here? You don't actually get a lot of instability. So extremes of mutational or extremes of genomic instability, they don't typically go together. So they're sort of mutually exclusive, but you do have a lot of cancers that fall in between, have some mutational rate and some levels of genomic instability. So what I call the idiopathogenesis and mutational signatures that are coming out a lot with these new genomic studies. Well, So mutations can be the consequences of many different processes, the intrinsic infidelity of DNA replication, where you have incorporation errors that just accumulate over the life of the organism or human. You have exogenous mutagens such as ionizing radiation, UV exposure, tobacco carcinogens, etc. Endogenous mutagens that occur through metabolic byproducts, oxidative stress, enzymatic modifications, and then you have your defective DNA damage repair in patients with hereditary breast and ovarian cancer syndromes and Lynch syndromes. All of these etiologies leave their signatures across the genome, and some very smart people have come up with bioinformatic ways to categorize this in large-scale genomic studies, and I think this is going to be a very particularly helpful outcome here for genomics and in applying it to individuals. Not going to get a whole lot into the bioinformatics, but basically there's 96 different combinations of how these mutations can be added up. And when you lay out all those combinations and tally up the mutations that occur in many cancers, you can get beautiful figures such as this and publish it in Nature. And they call these mutational signatures. 21 signatures have been developed or validated in cancer. It's likely more will be discovered. The most common one in this particular study was age-related spontaneous deamidation of cytosine. It was present in about 60% of cancers, probably not very surprising, but here are the age-related signatures here. The DNA damage repair deficiencies do produce specific signatures here. You can see the BRCA and homologous recombination deficient cancers, the deficient mismatch repair, and those polymerase epsilon mutated cancers. So having an ability to sequence a patient's cancer and then be able to say there's a widespread DNA damage repair pathway going on and we can detect that in that patient's individual cancer is really going to be very important in the coming years. Also, a highlight from the genomic study, something we didn't appreciate with traditional molecular biology techniques. Genomic studies have found an enzyme called ApoBec, and it mediates mutagenesis in about 15% of cancers, and this is really not well appreciated previously. And a little bit about ApoBec, it was evolutionary conserved cytidine deaminase, and it normally has a role in protecting cells against viral infections. But for whatever reasons, it's become altered and dysregulated in about 15% of cancers and has a spectacular signature that it leaves across the genome. Some signatures that they found, they don't really know what causes it, so this is an opportunity for discovery. Some commercial vendors are beginning to report these signatures for their research cases. I'm anticipating that we'll get the DNA damage repair signatures out as a routine here in a couple years. So just to briefly touch on viral-induced carcinogenous, oncogenic viruses do typically integrate their DNA into the host genome and lead to inactivating the retinoblastoma protein and P53 as their mechanism of oncogenesis. Some that don't integrate are typically things like HCV, hepatitis C virus, create inflammatory states that can lead to hepatocellular carcinoma. And importantly, many of the genomic profiling assays currently available don't report the viral etiologies of these things, so you're going to get something like a head and neck cancer that we know is HPV associated, and you're going to get a genomic sequencing result that says there's very few mutations there. So it's always important to keep in mind the particular etiology of the cancer you're sequencing. So what sorry for the typo there, but it should be fundamentals, concepts of targeted therapy. And these are things that I consider to be very important for looking at precision medicine and talking about the individual patient's tumors. So the really important thing is the principle of oncogene addiction. This is the phenomenon whereas a tumor remains dependent upon the sustained expression and activity of a single oncogene for its neoplastic progression, despite whatever else mutations occurs. It would follow that inhibition of that oncogene, you would produce a tumor response there. There's lots of caveats, such as mutation resistance mutations. Excuse me. So this principle of oncogene addiction has really allowed for rational drug design and targeted therapeutics to be developed. Well-known examples have been cited many times tonight, such as EGFR in lung cancers, HER2/neu in breast cancers, BCR-ABL, and some of the chronic leukemias. So here is a signaling cascade. This is a transmembrane receptor here that transmits a signal, typically, that can be shut off, on and off, with a growth factor. When you get something like an activating mutation in EGFR in lung cancers, that pathway is completely activated, and that represents sort of the oncogene addiction. That cancer cell must have that pathway on to survive. Also important is synthetic lethality. Synthetic lethality arises when a combination of mutations in 2 or more genes leads to cell death, whereas if you only have one or the other mutated, it doesn't really do anything. To sort of diagram this out, here's a normal cell where you have gene A and gene B both being functional, and that's cancer, or that normal cell is alive. In cancer cells, you can have gene A mutated or gene B and still have a viable cancer cell and lead to cancer and progression. What happens with synthetic lethality, though, is you have to have gene A and gene B there, and that combination is said to be lethal. And when you can find those combinations, that would cause cancer regression. So the key here is finding what gene A and gene B is in these cancer cells and being able to induce synthetic lethality. And one of the principal examples of this is PARP inhibition in the BRCA-mutated homologous recombination deficient breast cancers. So PARP essentially works under the principles of synthetic lethality. Now, in contrast to oncogene addiction, synthetic lethality may be more important for targeting tumor suppressors. Tumor suppressors are typically inactivated in cancers, and therefore you can't really inhibit something that's already inactive. And so this principle of synthetic lethality allows for some tumor suppressors to potentially be targetable here during some research. So another very interesting and one of my favorite topics is somatic genetic evolution. So this is the process by which cancers gain additional mutations as that cancer cell population grows, and you can diagram this out with some purple benign cells, and the first cancer cell here is in blue, and over time that population grows, and you have a very heterogeneous mixture by the time someone presents clinically with a cancer. It's composed of multiple different clones. So you can have intratumoral heterogeneity in that primary cancer, and if one of these orange clones decides to metastasize and that process of genetic evolution continues on, you can get primary and metastatic tumors that are very heterogeneous and sort of indistinct and separate from each other genetically. It's important to understand what a founder mutation is with somatic genetic evolution, and it's the first mutation that causes the neoplasm to basically initiate. And we say that the blue mutation here would be the founder mutation. It should be present in all of the tumor cells, and therefore it should be clonal. The mutations that happen later here, such as the orange mutation, should be subclonal when you think about it. It's not a founder mutation. It's one of those that is not present in all of the tumor cells. And this is all important because mutational processes differ between early events that initiate the cancers and the later events that propagate the cancers. So early founder events, again, they're predominantly clonal and very homogeneous. They occur in these very well-defined genes that we know of, the EGFR in lung cancers, the HER2s in breast cancers. Environmental carcinogens likely play a greater role in causing these founder mutations to occur. And what happens early in many cancers is the genome doubles over. And so when we're talking about ideal candidates for targeted therapies, these founder mutations are actually ideal candidates because if you can inhibit one under the principle of oncogene addiction, you can affect the whole entire tumor cell. Whereas the later events may be more heterogeneous, and inhibiting a subclonal mutation is unlikely to produce a widespread response. So these late events are subclonal. They may be more related to intrinsic processes, such as the ApoBec-mediated mutagenesis. Very interestingly, so once ApoBec starts as a late event, it helps diversify the tumor clones, is what they've been finding in the latest research. Very interesting. Also, chemotherapy or therapy-related mutations typically occur very late on there. So take-home point is that, we were taught, early on, or the early formation of a cancer is where, you have an accumulation of mutations. What the genomic techniques are finding is that there are very distinct processes that begin a cancer and very distinct processes that lead to the propagation of that cancer. So you hear a lot about driver alterations. Well, what is a driver alteration? Everyone sort of has their own definitions, and you hear different remarks made against or for driver alterations. But if you broadly define them, a driver alteration is any genetic variant that provides a selective advantage to a neoplastic cell. So any of those cancer hallmarks, if you have a genetic variant, regardless of it's a single nucleotide variant, an indel, a translocation, if it somehow promotes one of the hallmarks of cancer, it could potentially be called a driver alteration. And those can occur in tumor suppressor genes, oncogenes, or even promoter regions that are non-genic, non-coding. So how do you take all this and actually act on it? Now there's a lot of debate, and many institutions have their own criteria, or such as the NCCN guidelines will publish their own actionable results for some of these genetic variants. They're mostly congruent. They center on FDA-approved indications, standard of care, and what's investigational. I like to sort of think of it, well, you start with a genetic variant, and if it provides a selective advantage, or you can call it a driver alteration, you can check all your guidelines, see if it's actionable. There's typically, we think of actionable mutation or variants as either sensitizing to treatment, resistance to treatment. And within resistance, you can have de novo mutations, which are present at the beginning without any treatment, or resistance mutations that evolve out after treatment. Also actionable, we can look at these mutations that show up on the profiling results and sometimes be able to identify germline variants and recommend the patient go see the genetic counselors for follow-up testing and counseling. Just because something isn't actionable doesn't mean it's not a driver alteration. Again, if it promotes one of the cancer hallmarks and provides a selective advantage, it could potentially be called a driver alteration. Sometimes they carry prognostic significance, and other times they're a driver and we don't necessarily know what to do with them. You're going to get a lot of VUSs on some of these profiling results. A VUS is a variant of unknown significance, or it occurs in maybe a non-driver alteration. It's not actionable at this point in time. If you get a VUS, you should not act on those. Here is OncoKB. It's my favorite website. It's provided by Memorial Sloan. It's a freely available website to search and see if any of these results that you get are actionable. They've curated hundreds of genes, thousands of variants in multiple tumor types with lots of curated drugs. So you can see they have, level 1 is FDA approved, 20 genes, 10 genes are standard of care, and they have about 10 genes that provide resistance to treatments that they've curated. So actionable consequences of DNA damage repair deficiency. So homologous recombination deficiency has been mentioned a lot tonight. Cancers that arise as a consequence of homologous recombination deficiency show, three characteristic types of DNA alterations: loss of heterozygosity, telomeric imbalances, and what some of the research is calling large-scale state transitions. This is essentially genomic instability. And regardless of whether the mutations in BRCA are inherited as part of the germline or somatic, these homologous recombination deficiency cancers are highly correlated to these BRCA mutations. And so these are typically triple-negative breast cancers and the high-grade ovarian serous. And as mentioned earlier, the PARP inhibition, as it functions through a synthetic lethal interaction, is one of the treatments for these homologous recombination deficient cancers. A little bit about the mechanism here, yes. Since the PARP1 functions in the base excision repair to repair single-stranded breaks, if you inhibit that enzyme, a lot of single-stranded breaks actually become double-stranded breaks, and it overwhelms your already deficient homologous recombination system and leads to apoptosis. So mismatch repair deficiency, you get a lot of questions clinically about mismatch repair. These are equivalent to MSI-high cancers, cancers that arise as a product of defective mismatch repair. Well, we call them microsatellite unstable because microsatellites are repetitive DNA sequences, and when you can't repair mismatches, there's expansion of these repetitive sequences, and that's why we call them, they're microsatellite sequences, and so that's when they become unstable, that's why we call them that. These mismatch repair deficient cancers have very high rates of mutations because the replication errors are not being repaired, and this inherited form is causative for Lynch syndrome. So it's important, really, because FDA did approve a drug, immune checkpoint inhibitor, pembrolizumab, for any cancer that is deficient in mismatch repair or MSI high, regardless of the histologic type. So we can screen for mismatch repair deficiencies by two primary methods, immunohistochemistry for the actual proteins, or we can do the molecular diagnostic test, which is shown here. This is our capillary electrophoresis, where the normal tumor tissue, here you see increasing size fragments, and these nice peaks are the intensity. And you can see in the tumor here, you still get these benign peaks, but you also have a new set of peaks, and that's why it's unstable. So there's a high degree of overlap between these two techniques, but they really should be considered complementary. Another actionable consequence of DNA damage repair deficiencies, well, if you get a lot of mutations and we can measure it, we can call it a total mutational burden. Here's a TCGA plot. That's the Cancer Genome Atlas, has categorized hundreds, if not thousands, of cancers now. So this is the mutational rate here on this side, and the cancer type. Melanomas have the highest mutational rates out of any cancer, and the pilocytic astrocytoma comes in with some of the lowest. We really don't know what, in essence, causes a lot of these differences in mutational rates. Melanomas do, tend to occur in sun-exposed skin, and they accumulate a lot of UV damage, and maybe that's why. Not necessarily sure why there's a lot of variation in terms of the kidney papillary cancer having a very tight range. What we do know here is that these are the ultra-hypermutators that were mentioned earlier, and they occur in predominantly uterine and colorectal cancers. You also have the hypermutational type cancers and mismatch repair deficiencies showing up here, and this your run-of-the-mill endometrial type cancers with regular rates of mutation. So it's an interesting way that the total tumor mutational burden can help us categorize these cancers now on an individual basis instead of just saying in general these cancer types tend to have this amount of mutations. We can measure that now. And commercial laboratories are now offering this as a standard measure when you order large-scale genomic studies. And high tumor mutational burden is now an emerging biomarker for response to these immune checkpoint inhibitors. And the thought is if you accumulate a ton of mutations in the DNA, those mutations are eventually translated into protein, and those abnormal mutations turn into abnormal proteins. And that can elicit an immune response, especially if you apply an immune checkpoint inhibitor on it. So we're getting this on our routinely on our molecular profiling results now. It's been very helpful in some circumstances. So summary, genomic technologies have really provided a unique and new way to measure genome-wide DNA damage. There's a lot of new additional insights to cancers that genomics has provided us, and we can find many actionable events using genomic techniques to profile cancers. So next time we'll talk a little bit about the molecular diagnostics of how we detect variants and what pathological specimen and sampling factors are relevant for genomics.
Thank you very much. So our last speaker for this evening is Dr. Tina DeLilly, who is dual board certified in internal medicine and molecular genetics. She's one of a rare breed of physicians. We're fortunate to have her here. She's joined Hoag in 2012. She previously served as the medical director for the genetics program, but then also was more recently working, and we have some genetic high-risk programs, and she directs the pancreas cancer, so people that have genetic predisposition. We have a research protocol here that allows us to screen those patients to try to hopefully pick up pancreas cancer early. With similar leafomania, which is a germline P53 mutation. Patients have a wide variety of cancers. We have a protocol here for screening those patients. So she leads some of that. And she's going to talk to us next. Thank you.
All right, my turn. All right, so thank you again for attending. I hope that it's not raining on the way out. I will try to be brief, and I try to keep my information simple. I tend to think simply, and I hope that I can relay the information in a simple manner, and you walk away with some information that you can apply to your practices. So when we think of genetics and genomics in medicine, the principles and approaches permeate all areas of medicine. It's not just about cancer. Of course, we're going to be talking about cancer and how it applies to cancer, but some examples, and this comes from Thompson & Thompson, just like Surratt said, that's our Bible. That's one of the baseline books that we use when we go through our training for genetics and genetic counseling. And the first page that you look at, it talks about all the examples of where genetics comes into play. So, for instance, a pediatrician that's evaluating a newborn that has multiple congenital anomalies, obviously you're looking for a chromosomal abnormality or other underlying etiology. An obstetrician that is assessing a female, a woman, a pregnant woman that has advanced maternal age that has failed her initial screen that's come back concerning for chromosomal abnormality, is going under for an amniocentesis, and that sample is being sent off. That's another example. On the other hand, in the arena of neurology, for instance, someone will come in and they have a strong family history of Alzheimer's disease and they want to have genetic testing. ApoE gene is one of the genes that we typically test for. And the rationale for doing the testing is to do some long-term financial planning. On the other hand, an internist that is assessing a geriatric patient that has that has polypharmacy, and we typically think of polypharmacy as 6 to 7 medications on board that has multiple intolerances and allergies to medications, and you want to make sense of what's right for them. That's another reason to do genetic testing, pharmacogenetic testing on the CYP450 system. And then finally, a genetic counselor that specializes in hereditary cancers, that's assessing a woman that's coming in with a strong family history of breast and ovarian cancer. So some of the things that were touched upon before, I'm not going to hit on again. So basic definitions, germline, hereditary, and familial cancer. I will say that with familial cancer, those are the cancers that occur in families more often than we would expect by chance alone, and it can be due to genetic conditions, genetic component, or an environmental or lifestyle factor that hasn't been taken into account. I can tell you, for instance, in our pancreatic cancer program, we had a patient that has a strong family history of, breast cancer occurring on her, I think it was the paternal side, and she came in and she said, you know what, my whole family on that side was living in Torrance, and there are 3 oil refineries there, and the whole family is riddled with cancer on that side. So that's something to take into consideration. We can't prove it per se, but due diligence, we do the genetic testing, and and if nothing is showing up, we do have to take that into consideration. So the things I'm going to be talking about are the mechanisms of action, thinking outside the box more of how do we get to cancer, what are the reasons to develop cancer. So whether they're chromosomal abnormalities, structural, meaning autosomal, or sex chromosomal abnormalities, your traditional Mendelian disorders, whether it be autosomal dominant, recessive, or X-linked. I'm not going to be talking about X-linked. There aren't too many of them, but going to hone in on dominant and recessive. And then you have your microdeletion syndromes, telopathies, microRNA disorders, and overgrowth syndromes. So going on to structural chromosomal abnormalities, the autosomes. So you got your crash course on what a karyotype looks like. So we're looking at trisomy 18 right over here. So that's Edwards syndrome. So the majority of these babies, they have multiple congenital anomalies, over 130 different conditions, whether it's heart defects or hydrocephalus. All these things have been enumerated and described for these babies, and the majority do die, and only 10% survive by 1 year of age. So when we talk about cancer for these babies, it's more, they're young, they're 1 year of age, and it's Wilms tumor, nephroblastoma that we see in these babies. Trisomy 21, that's your Down syndrome. So with this one, we see ALL and AML. So acute lymphoblastic leukemia and acute megakaryocytic leukemia, those are the two forms, and it's more of the AML that we see than the ALL. There's a 20-fold increased risk for ALL versus AML, which is actually relative risk of 400, very high. And we do see that in infants that have transient myeloid proliferation, they do may or may not go on to develop AML. Interestingly enough, adults, Down syndrome do go on to become adults. They do typically die in their 50s or 60s. There are cases of them living longer. They have Alzheimer's. They do live. We don't see cancer in the adults, interestingly enough. All right, so when you talk about the sex chromosomes, we're talking about things like Turner syndrome and Klinefelter. Turner syndrome, it's so you have a girl here, she has Turner, so they have short stature, they have the webbing of the neck, they have a wide chest with wide spaced nipples, they have premature ovarian failure, and so the cancer that we see associated is dependent upon this SRY gene, that if they have that or if they have gonadal dysgenesis, there's a higher risk of cancer associated with that, specifically with gonadoblastoma. Likewise with Klinefelter syndrome, so that's your extra X chromosome, there's an increased risk for male breast cancer, 20 to 30-fold increased risk, and mediastinal germ cell tumors. So when we talk about germ cell tumors, only 5% of them are seen outside of gonads. So mediastinal is very rare, but we do see it with Klinefelter. Okay, Mendelian forms of cancer susceptibility. So when you look at it broadly, inherited cancers, typically when you think of the, colon, breast, and all that, hereditary cancers, just like Jeannie said, is, no more than, it's about 10%. But when you look at it from birth to adulthood, pediatric to adulthood, it's, it's a broader range of 1 to 60%. The more, oops, I have a typo. For the more common tumor types, the inherited fraction falls in the range of 1 to 10%. And the take-home message is the more rare the cancer is, there's a higher likelihood of it being inherited. So we're talking about your sarcomas, optic gliomas, adrenocortical tumors, retinoblastoma, as high as 60%. Tumor suppressor genes, we talked about that. That's something that we see, and it's more associated with the autosomal dominant conditions and the oncogenes, right, the proto-oncogenes. So concepts of penetrance, expressivity, anticipation have been already discussed. De novo mutations, that means that it wasn't inherited, it happened spontaneously, and that does happen, and we do see that. Okay, so when we talk about autosomal dominant conditions, we're talking about multiple generations that are affected with variable proportion of de novo mutations. Certain tumor types do cluster the patterns, right? So for BRCA1 and 2, we know, breast, ovarian, melanoma, pancreatic cancer, those are the clusters that we look for. There's variable penetrance, and you do see unaffected mutation carriers. So I've just listed a few of the conditions that are associated with proto-oncogenes. So MEN2, you have a RET mutation. And so again, going back to the whole, what do we call mutation? I may say mutation one time and then a pathogenic variant another time. So The newer nomenclature, the newer way of saying it is pathogenic variant. We try to stay true to that, but I do slip and say mutation. I'm used to it. It's shorter. So RET mutation, you see ME, it's associated with MEN2. So MET, that's another one. Interesting ones that I'm going to be talking about are your RASopathies, the HRAS and the KRAS. So Dr. Braxton talked about the RAS, pathway, and so the somatic mutations that we see associated with that has a genetic signature. We do see actual germline mutations that are associated with syndromes, and I'm going to talk about that a little more. So, and then again, ALK and EGF are associated with certain cancers as well. Okay, autosomal recessive conditions. So they're rare and they're associated with childhood cancers. And the genes that are involved or affected are either DNA repair genes or checkpoint response genes. Bloom syndrome is one of those. Bloom gene, the BLM gene, it's on chromosome 15q26.1. It is seen in Ashkenazi Jewish population with a carrier frequency of 1 in 100. There's a hot spot in that gene, and it's a deletion and insertion mutation. I do have the 2881 wrong, so for those of you who caught it, I've noticed it, so but that's the hot spot. So if someone comes in and they're of Ashkenazi descent, there's a specific hot spot within that gene that is screened with. So what do you see with that? You see intrauterine growth retardation, you have pigmentary changes, and then as you see on this child, you have this butterfly telangiectasia rash that is photosensitive. They have immunodeficiency that sets them up for long-term risk of infection that sometimes leads to death, and azoospermia. The risk of cancer is colon, breast, melanoma, among other types of cancers. Okay, another one on the list of autosomal recessive is hereditary ataxia telangiectasia. So what you see is this ataxia, so cerebellar ataxia, and so you see this girl, she's wheelchair bound, and that's not a surprise. The majority are wheelchair bound by their teenage years. You have telangiectasias of the conjunctiva, which you see up in this other picture. Immunodeficiency, IgG, IgM, IgA deficiency with infections. They have lymphoma, leukemia, and solid tumors. And their cells are sensitive to ionizing radiation, and so the dose of radiation needs to be decreased. Likewise, they have increased sensitivity to chemotherapy, so they do have toxicity associated with that. So I didn't provide a slide for those that are heterozygotes, which are the carriers. Carriers of the ATM gene, which is the gene that's involved in this condition, are at risk for cancer, and we typically cite a fourfold increased risk for specific cancers such as colon, breast, and pancreatic cancer is on there as well. All right, microdeletion syndrome. So we're not talking about the whole chromosomes anymore being duplicated or deleted. We're talking about a segment of the chromosome, and so a few genes within a small region that cytogenetically it can be seen that's deleted, and we call that a microdeletion syndrome. And it's typically associated with deletion of tumor suppressor genes. So there's one condition called Weger syndrome. It's Wilms tumor, aniridia, genitourinary abnormalities, and mental retardation, which we now call intellectual deficiency or dysfunction. So the two hotspots are the two genes that we think of and we worry about are WT1, Wilms tumor, and genitourinary abnormalities associated with it, and the PAX6 gene, which is associated with aniridia. Dennis-Drash syndrome is a missense or nonsense mutation that you specifically see in that WT1 gene, and it's associated with 90% risk for Wilms tumor. So the ones that have Weger itself they have a 20% lifetime risk for kidney failure. And so because they're at risk for Wilms tumor, we start the screening by abdominal ultrasound at birth, and we go up until age 8, just so that we can find the Wilms tumor. Another condition that kind of sort of is under a microdeletion syndrome is retinoblastoma. And it's in about 3 to 5% of cases, you see a germline deletion of 13q14.2, which contains that RB1 gene. It's a cell cycle regulator, and it's expressed in all cells. 1 in 20,000 children are affected. It can be unilateral or bilateral, and those that have bilateral retinoblastoma, 80% of them have a de novo mutation and don't have a family history. If it is untreated, it leads to blindness, and it's fatal once it spreads outside the eye. Long-term survivors, so if they get past that hurdle and they get treated, long-term survivors have a high rate of secondary malignancies, including bone and soft tissue sarcomas, uterine and lung cancers in adulthood. So they've done studies in 68% of the patients in the study had second primary malignancies through age 50 with epithelial tumors. All right, this is one of my favorite areas, and I don't get to talk about too much, but we talked already about on the somatic side, right? So on the tumor side, we know there's this KRAS, tyrosine kinase pathway that is very important. It's one of the hallmarks for cancer development. So, for instance, we know with KRAS that's mutated, and David, if I'm wrong, you can correct me. KRAS is, we see that, in 90% of pancreatic adenocarcinoma on the tumor side that we see, that it's mutated. Also, it's seen in colon cancer and in lung cancer. BRAF, I believe, is with melanoma. We see that, and NRAS as well with melanoma. So there is a signature there from the somatic side. Well, you can have germline mutations within this pathway as well. So if you have, Costello syndrome, and I'm going to show you pictures on the next slide. Costello syndrome is one condition where you have a mutation, a germline mutation in the HRAS. Cardiofacial cutaneous syndrome is another one, and you can have mutations in KRAS or MEK1 or MEK2 or BRAF. And so Costello syndrome is associated with, I believe, with bladder cancer, and cardiofacial cutaneous syndrome they think is associated with ALL. We know I think you're more familiar with neurofibromatosis, right? So NF1 definitely is associated with malignancies, whether it's nerve sheath tumors or gliomas or meningiomas or pheocytochromas, it's there. Noonan syndrome is another condition that we see, and you have von Willebrand disease associated with that one. So, interestingly, some of them don't have cancer associated with. Legia syndrome is one that you don't see cancer with. And so here, you have cardiofacial cutaneous syndrome here on the left, and then Costello on the right. You can see that they have coarse facial features, and they look similar, so it's sometimes hard to tease them out when they come in and we do a genetic evaluation, and it's not until we do the genetic testing that we can figure out what it is that they have. Noonan syndrome is sometimes called the male Turner syndrome. You can see that that's short neck, webbed, and they have some, the dysmorphology is there. This is a very prominent case of neurofibromatosis, where you see these neurofibromas. The woman is riddled with them. All right, switching gears a little, overgrowth syndromes. So there are about six of them that we learned when we went through our training, and Beckwith-Wiedemann syndrome was one of the hallmark ones that we learned and understood well. The genetics of it is very complicated, and they test us all the time on this, and it's not simple. So it's hypermethylation, hypomethylation. There are mutations in there. There's uniparental disomy. There are different ways to get to Beckwith-Wiedemann syndrome. And so the hallmarks of this syndrome they're large babies. So they're macrosomic, and they have large tongues, macroglossia. They have organomegaly and hemihyperplasia. They have an overall risk for Wilms tumor of 2 to 5%, with a hepatoblastoma risk of 1 to 2%. If hemihypertrophy, which is, whether one leg is bigger than the other, or, that's what we typically look for. We do a lot of measurements when we see them as babies, or if they have hepatomegaly, there's an increased risk with tumors. So just like Wager, we start the screening for kidneys by ultrasound and alpha-fetoprotein for hepatoblastoma right at birth, and we do it every 3 months up until age 8 years. All right, another area is telomere disorders. We call them telopathies. So just a little about the structure of the telomere. So you have those tagged nucleotide repeats that forms a T loop, and then you have these proteins, this capped protein, these proteins on top, and then you have the enzyme complex here that produces telomerase, which is involved in keeping the telomere, the tips of the chromosomes, intact as it goes through replication. So dyskeratosis congenita is the one condition that we talk about when we talk about telomere disorders. It's associated with very short telomeres, less than 1 percentile as compared to age-matched. There's a clinical triad of nail dystrophy, oral and buccal leukoplakia. They have hyperpigmentation of the skin. They usually don't live beyond the 3rd decade of life. 20%, the malignancy that you see associated with 20% AML, you see head and neck cancers in young adults. Morbidity and mortality is associated with bone marrow failure, immunodeficiency, and they have pulmonary complications as well. Okay, so microRNAs. So microRNAs, when I went through my training, I don't think we even had it. So I think it was in the 1990s that we started looking in and figuring out, these are non-coding RNAs, and their job is to regulate other RNA, messenger RNA, that, so it suppresses the protein, the translation of a protein. And so this is just a pathway showing you what happens, that the microRNA binds to the messenger RNA and it suppresses the activity. So DICER1 is actually an autosomal dominant condition, and it has incomplete penetrance with variable expression. So young children who carry this variant undergo, I'm calling it a variant now, so I go back and forth between mutation and pathogenic variant, undergo screening focused on surveillance of the pulmonary cavity. They're at risk for goiter, nephromas, ovarian sertoli type ovarian tumors, Wilms tumor, rhabdomyosarcoma, pituitary blastoma, and pineoblastoma. So when you pull all these mechanisms together, you get cancer, and you have different mechanisms. So trisomy, when we talk about Wilms tumor, we know trisomy 18, those long-term survivors, they're not really long-term, but they are at risk for Wilms tumor, and it's typically bilateral. Wager, which is a microdeletion syndrome we talked about, has, they're at risk for Wilms tumor, and then Dennis-Drash and Perlman syndrome, Beckwith-Wiedemann. So there are different ways to getting to one cancer, but it's important to figure out why. And remember, the more rare the condition it is, the more likelihood that it is genetic etiology. And so it's important to figure this out. So that's it for now. Part B is going to be about more on hereditary and familial cancers, testing approaches, panel testing, and how do we infer germline findings from tumors, and on selected cancer patients, the germline findings. So.